bank. Okay, go gene bank gene. MARCO. Okay.
Yeah, that's it. Macrophage receptor.
Macrophage receptor with a collagenous structure is a mouse.
Yeah, mouse.
Or human mouse, right?
Mouse.
Okay. This is the gene. Okay, let's find the gene sequence.
It's a big gene.
It's big. You want to target? Okay, genomic sequence. Great. Use. I have to use this one. The vector. The new system.
LY112. That's a genetic.
Yeah, I just want to open from this where I have two Windows system. I want to open genomic sequence. That's the one. Okay, that's a gene. All right.
Where did you stay? How did that get into there? So
It's just automatically. That's a software.
What software is that?
Vector NTI.
Oh, okay. Is that the vector? Is that the one that costs a lot of money?
Yeah.
Okay.
It used to be so 2003, 2004, it's a lot of money, and then 2006 it's free. It's free for 5 years probably. Okay. Then a lot of people begin to use it, and then 2010 I believe it began to charge money. It's $2,000 for static license. That means forever. You pay $2,000. You can.
It's a nice program. I can see it puts a nice genomic structure and everything.
Yeah, some other software probably you can find some free software. They can do similar function, but I've been using this for 10 years, so kind of familiar with this. So that's a gene. This is an exon. So depends on where do you want to target it. You want to knock out which exon, so we can find the potential gene, potential sequence.
Oh, this is sequence.
Yeah, this is sequence. So.
So, something that's would knock out the frame. Make it out of frame if it was gone.
Knock out one gene or one.
Well,
exon.
let's just say we want to knock out.
I can knock out the whole sequence. The whole gene. We can knock out the whole gene.
With CRISPR?
With CRISPR or without CRISPR. Probably without CRISPR, the efficiency probably a lot high also.
Well, how about just, let's say you want to knock out that one. The second
one?
Yeah. Sometimes the second one is good to knock out. Oh, I see. It highlights it.
I need to find, I don't know, some coding sequence. mRNA. Okay, that's the exon. Let's find the one, 2, 3.
So the start on the whole thing is 30,000, right? So the exon is.
Yeah, let me find the exon 2. Okay, this is exon 2. If you want to use your CRISPR to target this one.
Yeah, that's a small one.
Okay, we can find GG. Just look at GG, and this is 3G.
Yeah.
Okay, GG, the 19 base pair. Before this is a G.
How are you counting? What's counting here?
Here.
Oh, I see down there. Yeah, very easy.
It's not a G, so I think it's. Let me make sure.
It's a G on the other strand.
That's you have to. You look at the top. You just focus on. The top.
Oh, it has to be G on the same strand.
Yeah, you have to on the same strand. Let me double check how many base pairs need.
Well, if you move here, look. Here's 2 G's.
19.
Okay,
we can go down. Okay, from here.
I think it should be separated there.
Oh, this is good side. We can target this. We can just insert this sequence into the CRISPR. plasmid, and then make the plasmid, and then put the plasmid into ES cells, and then put the donor DNA. See,
What's the donor DNA?
like you can put slash marker, new myosin
Oh.
gene, whatever, to replace this one, to add a slash marker. So
And this might knock out this exon.
yeah, knock out this. You can even knock out the whole thing. But for CRISPR, I think
How would you knock out the whole thing?
just same thing. You use the homologous recombination, and put the homologous sequence this end, that end. Then, but for CRISPR may not be efficient. CRISPR usually knock out the place that the target. But if you knock out the whole thing, it's probably low efficient.
Yeah, okay.
Yeah.
All right. And you're not sure which will work, so you do more than one.
Yeah, you just find another place, GG and G, where you can find another gene,
Yeah, just
another exon. Actually, there must be some software. You just put the whole sequence into the software, then they can find the sequence.
right.
But
That's
it's not difficult. We can just use it manually. We can find another exon, 10823 to 11041.
what are you doing? Picking
I'm
another
looking,
exon?
picking another exon.
Like the
You
third
see,
one?
based on this is drawn. This is splice. So what is the splicing?
Splice out, yeah.
This is genomic DNA. So if you find exon, this is another exon. So you can just try the GG and GG. Okay, there are 2 G here. Then from here, 19 base pair. This is 20. No, this is not good. We'll try. I'll try this. 19. This is not good. This is G. Oh, this is another good site for CRISPR targeting.
Are there 2 G's there at the beginning? It looks like a GC.
Huh?
Is that 2 G's or GC?
This is 2G. 2G here.
Oh, 2G there. And
Yeah, just need 1G. So this is 19 base pair GGN.
From which one? From which G? Which G is the beginning G? Those 2 G's?
Yeah, this 2G is the beginning G.
Oh, those 2 G's.
Yeah.
And then you go 19 after that. And it
Yeah.
should be a G.
Yeah, then you find a 19. This is another G.
Now, how can this be so specific? Because this thing came up here. How again is it going to be specific for that region?
Yeah,
Because you see, you find them all over the place.
yeah,
TG and then 19 another G. How can it target this one versus all the other ones?
this is 19. This is unique for this gene, right?
Okay, right.
This is 19.
Right.
base
Okay,
pair.
that's that part. All right. Now, what about the making it more specific?
More specific.
Well, I mean, this comes up. Yes, it's unique for this gene, but there's other genes.
That's the off-target issue. So if other gene has same sequence, it target also.
But
It will
they definitely will, right? Because you
Yeah.
can find it everywhere.
That's so the more actually the 19 base pair only 10 or 13 is critical. The
Yeah.
other 5, 6 or 7 are not critical. So actually you only have 13 or 12 base pair. If other gene has same, then it will target. So that means the off-target issue is a big issue.
Right. But what do you do about that? How do you solve this?
Yeah, that's the thing. That's a two CRISPR. That's you based on this paper. You use the mutant Cas9. Use the mutant Cas9. It won't cut or only make a nick. So you make a nick, then you make a nick here. You have 2 CRISPR, 2 guide RNA.
Yeah, it
So
says.
then it will be more unique, right?
How do you get the unique? Okay, yeah. Where's the second one?
Then you have to find. So let's say we just find one here.
Right.
Then we need to find another one closer to here. If we don't find another one, then we won't use it.
Oh,
So
I
we
see.
have to. We find one here. That's 19. And then we find another one here. Okay. Then we can use 2 CRISPRs to target this this side.
And I guess the intervening sequence
The intervening sequence is important.
is probably, that's important. And it's not that those two, even this intervening sequence won't come up a lot, right? And can you blast it? What about, say, you get something that you think is good and you see if it's anywhere else?
Yeah.
Can you do that?
That's still, If we can blast. If you have target sequence, two target sequence, then you can make blast.
Can you do that real quick? Because you're good at this.
We can.
Let's see if this target sequence.
We can let's do it. Okay, that's we just found one side.
And see how often that thing plus the intervening sequence comes out.
Let's say this is a target sequence.
Okay, right.
I just assume
Right.
it's not, but
okay,
so
right. I understand.
Put our gene here. You show your mouse. It will tell you how many places it can match. So take a while. See. Okay, 100%. Again, it's not very bad. So this one is 83. So 2 to 16, this will be same. So we put a 19 base pair probably. So it's 2 to 16 is exactly match another gene.
Which one? 2 to 16?
We put when we did the blast, this is 19 base pair, right? So from this 2 to 16, it match another gene.
You mean there's no perfect match though?
It's not right. No perfect match is the gene.
That's not so bad.
It's not bad. It's, this actually is.
That's good enough, or for
you see, Mark 1 is pretty unique for this one.
Oh, well, that's all right.
It's ready.
Oh,
So
I see.
It probably can still target this side. That's a problem.
totally different.
So, but if you put 2 in,
Yeah, you put 2, then it target one CRISPR target this gene, another CRISPR target another gene, but you have two CRISPR simultaneously bind together, so it won't target. It will only make a nick. When the nick, the cell will self-repair the nick. So then you can solve the off-target issue.
Oh, and does it cut out the intervening thing?
What?
How does it repair the nick?
But if you put them together, it will cut.
Yeah.
It will. It will.
Then the
cut
Cas9 comes on, cuts out the intervening thing, and disrupts the gene.
It will cut the intervening gene, and then the repair the cell system repair system will make a mutation.
Right.
It's a different repair mechanism.
Right.
You make a nick, the cell can self. It won't make when the cell make repair the mutation. It won't make a mutation. But if you cut both the cell mechanism, they want to repair too. It's different mechanism. It will introduce mutation. So.
Can you go back to that site where you picked the nicking? Sounds important. Which site was it that you saw the nicking thing? I think it was whatever site this thing is on. The CRISPR site.
The ad gene, you mean?
Yeah, the ad gene.
Oh yeah.
So it had nicking as a
Oh, I
thing.
close it. Okay. Oh, it's in this side. It's 2 different systems. Yeah, nicking.
So
See?
this is important.
Yeah, this is important. This is
Don't just
important.
cut, but nick. The idea is to nick 2 places close by. This is what the people use. Can you go to open this up and see what's hot there?
Open this.
Yeah.
Open already.
This is very hot, right? So what's a hot way to nick? Down here.
I don't know. Maybe this is the nick. I probably just open again.
So this is the thing to do, right? If you want to take a gene out,
Yeah,
and relatively
if you wanna,
specific.
Yeah, right now we don't use the wild type of Cas9 anymore. But based on your purpose, you can still, because wild type it should be more efficient than nick. A cut double strand it should be more efficient.
And the D10A, right?
That's D10. That's a mutation. That's the site. That's the 10th amino acid mutated from D to A.
Oh, I see. Right,
That's
right,
D10A
right.
mutation.
And which one's hot down here?
I don't know. Check. Oh, should be this one. These 2.
Okay. So, and why are they hot? Codon optimized, D10A, mammalian.
You can read the paper. This is the
I
lab. You can open and then check the reference publication. You can read, see what's the benefit, what's the advantage.
Oh, I see.
Yeah.
All right. Oh, Wataru Fuji Lab plasmid.
Yeah, Japanese, I believe.